Cassava roots, peels and leaves should not be consumed raw because they contain two cyanogenic glucosides, linamarin and lotaustralin. These are decomposed by linamarase, a naturally occurring enzyme in cassava, liberating hydrogen cyanide, HCN. Cassava varieties are often categorized as either sweet or bitter, signifying the absence or presence of toxic levels of cyanogenic glucosides, respectively. The so-called sweet, actually not bitter, cultivars can produce as little as 20 milligrams of cyanide, CN, per kilogram of fresh roots, whereas bitter ones may produce more than 50 times as much, 1 gram/kg. Cassavas grown during drought are especially high in these toxins. A dose of 25 milligrams of pure cassava cyanogenic glucoside, which contains 2.5 milligrams of cyanide, is sufficient to kill a rat. 40. Excess cyanide residue from improper preparation is known to cause acute cyanide intoxication, and goiters, and has been linked to ataxia, a neurological disorder affecting the ability to walk, also known as konzo. It has also been linked to tropical calcific pancreatitis in humans, leading to chronic pancreatitis. Symptoms of acute cyanide intoxication appear 4 or more hours after ingesting raw or poorly processed cassava, vertigo, vomiting, and collapse. In some cases, death may result within 1 or 2 hours. It can be treated easily with an injection of thiosulfate, which makes sulfur available for the patient's body to detoxify by converting the poisonous cyanide into thiocyanate. Chronic, low-level cyanide exposure is associated with the development of goiter and with tropical ataxic neuropathy, a nerve-damaging disorder that renders a person unsteady and uncoordinated. Severe cyanide poisoning, particularly during famines, is associated with outbreaks of a debilitating, irreversible paralytic disorder called konzo and, in some cases, death. The incidence of konzo and tropical ataxic neuropathy can be as high as 3% in some areas. During the shortages in Venezuela in the late 2010s, dozens of deaths were reported due to Venezuelans resorting to eating bitter cassava in order to curb starvation. Societies that traditionally eat cassava generally understand that some processing, soaking, cooking, fermentation, etc., is necessary to avoid getting sick. Brief soaking, for hours, of cassava is not sufficient, but soaking for 18 to 24 hours can remove up to half the level of cyanide. Drying may not be sufficient, either. For some smaller rooted, sweet varieties, cooking is sufficient to eliminate all toxicity. The cyanide is carried away in the processing water and the amounts produced in domestic consumption are too small to have environmental impact. 37. The larger rooted, bitter varieties used for production of flour or starch must be processed to remove the cyanogenic glucosides. The large roots are peeled and then ground into flour, which is then soaked in water, squeezed dry several times, and toasted. The starch grains that float to the surface during the soaking process are also used in cooking. 49. The flour is used throughout South America and the Caribbean. Industrial production of cassava flour, even at the cottage level, may generate enough cyanide and cyanogenic glycosides in the effluents to have a severe environmental impact. Please like, share and subscribe.